So for the next two chapters, we're going to talk about the human immune system, and there's two distinct functional divisions. So we'll start with the first one in this lecture. So the human immune system is divided into two distinct systems. What we call the innate system is non-specific, meaning it can't tell the difference between bacteria or virus. It just knows that it's something that is non-human, essentially. And so these are the things we're born with: physical defenses like skin and mucous membranes. And then we also have an adaptive system, which is the one that gets improved over our lifetime as we get exposed to things. And that's why we call it the very specific immune system, and it's also acquired. So this is what we'll talk about in chapter 13. So the general function of the immune system is basically to constantly surveil the body and look for foreign substances or foreign materials. So we do this by having surface receptors on our white blood cells, which are essentially the immune system, and whether they have affinity or not for substances that come their way or cells that come their way. So those surface markers on our cells, what we call self, we would be tolerant to those surface markers, right? So they call this self-tolerance. So when a white blood cell comes along and matches up its receptors to surface proteins of good, normal self-antigens, then they should basically ignore those cells. It's only when they come in contact with foreign particles, so those antigens on the surface of bacteria or viruses or any other pathogens, would then be recognized as a foreign substance by the white blood cells, and would then proceed with destruction of that foreign particle. So when we lay out the immune system, we usually think of it as a first, second, and third line defenses. So our first line defenses are those physical barriers that just keep everything out. Assuming it's something that is not fat soluble, or it doesn't have some method for boring right through the skin or mucous membranes, it should be kept out of the body. So when we think of the skin, it's not just the physical layering of the dead cells, but it's also the sebum on the surface of the skin, which makes it very acidic, right? Think fatty acids, acidic. We also have salt on our skin that is very inhibitive to certain organisms, especially things like yeast. And then we also have the normal microflora that we met back in chapter 11. Those organisms that belong on the human body, when they ferment and they digest the molecules on the surface of the skin, they produce acids, and that also contributes to this acidic nature of the skin. If they're able to bypass that first layer, then we have things like phagocytes, and we're going to talk about things like complement and interferon. We have inflammation and fever. All of these are very non-specific. They will just respond when something non-human comes along. And then in the next chapter, we'll come back with those very specific, what I like to call special forces. So if you get past all of those initial non-specific defenses, then we're probably going to have to go chase them down with something like an antibody or a T cell, which knows how to find viruses and cancer and things like that. So one of the most important barriers, going back to our first line defenses, are mucous membranes, and especially those that line the respiratory tract, right? The respiratory tract is a very important portal of entry because it is open to the outside world. And so we have what's called the ciliary escalator that is located along the lining of the trachea. So anything that gets trapped on the mucus of that passageway will then constantly get washed up and out of the trachea and then swallowed and goes down into the esophagus and into the stomach, where the pH is very low, and that acidic environment can destroy most particles that come its way. So if something does get past the first line defenses, it's going to then come in contact with what we call second line defenses. And pretty much all of these next layer components are found in the blood, right? So things like phagocytes, they circulate in the bloodstream. We're going to have a series of proteins, what we call interferon and complement, and these can also travel in the bloodstream. Chemicals that trigger inflammation and fever will also be distributed all throughout the bloodstream. So recall what phagocytes are designed to do. They ingest and destroy foreign particles. And so we have several types of phagocytes that are actually residents in different parts of the body. So starting up here, kind of go clockwise. In the lungs, we have alveolar macrophages. Sometimes they're called dust cells. In the spleen, we have macrophages. One of their main jobs, of course, is to get rid of old red blood cells that can't manage to get through the filter of the spleen. But those macrophages in the spleen can also encounter and destroy foreign particles that come through in the bloodstream, because, of course, that's what the spleen does. It filters blood. In the kidneys, we have resident macrophages to help with whatever gets filtered out there. In the skin, we have dendritic cells. You may recall from AMP1, we usually call those Langerhans cells. We even have resident macrophages in joints all throughout the body. We have monocytes, which are circulating in the bloodstream. When they leave the bloodstream, we call them macrophages. Of course, the lymph nodes are really good filter areas, and so we have lots of macrophages stationed in those lymph nodes. We also have residents in the liver called the Kupffer cells. And then we even have phagocytes inside the central nervous system. Within the brain, we have microglia. And so because the blood-brain barrier is very effective at keeping everything out, it also keeps out the white blood cells that circulate in the bloodstream. So we have to have resident phagocytes on the inside of that barrier. So if you recall the process of phagocytosis, you have to have affinity with something to adhere to. So this is where, when we talk about the coatings on bacteria that keep them from being phagocytized, if they can't actually bind to the foreign particle, then they can't proceed with this process. So adhesion is key. Then they'll pull them into a phagosome. The phagosome then binds to the lysosome. And then that's where the powerful lysosome enzymes have contact with the particle and they get destroyed. Now, everything that is debris essentially will get eliminated from the cell, but we're also going to see that there's a step here where instead of just destroying everything about that particle, the phagocyte will actually retain some of its surface markers and do what we call antigen presentation. So we'll hear more about antigen presentation as we go. So one of the antimicrobial proteins we're going to talk about are the interferons. And this is a large family of chemical messengers that are released by our tissues and even some of the immune system cells. And their job is to basically regulate the immune response. And so the ones we're going to talk about specifically are those that are released by our tissue cells when they are infected by a virus. And so their job is essentially to limit the spread of the virus. They don't actually kill the virus. Of course, viruses aren't technically alive or dead, but all they can do is interfere with the viral replication, which is how they get their name. And so they are triggered, they get secreted when our host cells get infected by a virus. The presence of that foreign nucleic acid, whether it's DNA or RNA, will trigger the host cell to secrete interferons, which are just little protein messengers, and they only go as far as their neighbors. Okay, and so the neighboring cells have receptors for these little interferon messages, and when they receive that message, it causes the healthy neighboring cell to produce little proteins that stay inside that healthy cell called antiviral proteins. And those antiviral proteins then bind to and block ribosomes, which of course we know is where the translation step of protein synthesis occurs. And so because no matter what kind of virus you are, if you recall from chapter 5, if you're a virus, you have to spend some time at the ribosome so that you can make your own proteins, whether it's a capsid protein or an enzyme of some sort. If the ribosomes are shut down, then viruses can't replicate. So really what interferons do is just buys time for the immune system to come and find that cell that's harboring the virus and kill it with other means. But keep in mind, if you shut down the ribosome, that also means that the host cell can't make any proteins. And so it's really sort of a self-inflicted thing that happens when we have a viral infection. We get symptoms like malaise, muscle aches, fever, all those things are actually sort of triggered by the cell shutting itself down. And so this message can really only last for about 3 days. Otherwise, those cells would die if they couldn't continue to make their own proteins. They would stop metabolism, their enzymes would not be able to be made, things like that. And so because this shuts down ribosomes, these are really most effective against RNA viruses. So if you recall, if you come in looking like something like messenger RNA, you're a virus that gets to go straight to the ribosome and start copying yourself. And so obviously if it's something like a DNA virus, because those guys can park it in the DNA and stay there for years without actually replicating themselves, a message like this that only lasts about 3 days is not going to do much good. So obviously interferons are not the only way we have to deal with viruses, because it's not going to be effective against all kinds of viruses. So here again is what it looks like. So a virus infects a host cell. Once that viral DNA is present, it triggers the interferon genes that, by the way, all of our cells have the capacity to make interferon, and all of our cells have interferon receptors. So obviously if this initial cell that gets infected was, for example, a respiratory system cell, and the virus is influenza, then the neighboring respiratory cells are the ones that are most susceptible to this particular virus. And so it makes sense that you would want to warn your neighbors that a virus is in the neighborhood and is probably going to replicate and come your way. And so it's those healthy cells that get the warning, basically. That's what the interferon is. It's a warning. So that initial cell that released the interferon is going to die, right? As soon as that virus finishes replicating, it's going to burst out and probably kill the host cell. That virus will then hopefully go next door and find a new susceptible host cell. And when it tries to replicate there, the antiviral proteins that have been made in that new cell have shut down ribosomes. And so it basically stops the replication or interferes with the viral replication. So it doesn't really target the virus directly. It just makes life really hard for that virus. And it buys time for the T cells, which would know how to come find this cell that's harboring a virus and destroy it. So interferons can only interfere with viral replication. But this is important because one virus in can mean millions of viruses on the way out with just one generation time. So it's really important that our body tries to get ahead of viral replication because they're so effective. So because interferons are so good at interfering with viral replication, there's been a lot of research into developing drugs that involve interferons. Now, the problem is interferons are just tiny little proteins, and the body has such an efficient clearance rate at the kidney and the liver that it's hard to keep these little proteins in high enough concentration in the bloodstream. The other problem is that interferons, there's so many different kinds of interferon, and every cell in your body has the capacity to hear the message of interferon. So obviously, if you're putting interferons in the body, you don't want to shut down ribosomes all over the body, all at the same time, and then you'd have to keep this drug on board for, long enough to actually find the target cells and things like that. So it's been really problematic, and most of the infections that they have developed interferon for have not been effective. There are a few that they have found some effectiveness, so hepatitis B and C, Kaposi sarcoma, if you recall, that's a herpes 8 infection that's typical of HIV/AIDS patients. So, there's 50 years of research on interferons, and we still don't have more than just this little handful of infections that can be targeted with interferons. So if interferon is targeting viruses, complement will target bacteria. And so this is basically a series of proteins that are produced in the liver that circulate in the bloodstream in an inactive form, much like the blood clotting cascade. And so once you turn the first one on, it turns on the next one, and that one turns on the next one. And basically, once you turn on this cascade, the end result is going to be to create what's called a membrane attack complex, which causes lysis in the target cell. And of course, the target cell here should be a bacteria. And so because gram-negative bacteria have such a thin cell wall, these little MAC proteins can find their way past the cell membrane, past the outer membrane, past the cell wall, and into the membrane of gram-negative bacteria. So we have three ways that this can be turned on. One way is that if there's an antibody already present that is bound to a bacteria, in this case they're calling it the antigen, it's typically a bacteria, then the complement can stick to that antibody and progress through the cascade. Another way is that the complement proteins themselves stick directly to the surface of the bacteria, or another pathway where the complement sticks to these little sugars on the surface of certain bacteria called lectins. And so no matter how you turn it on, you end up with this cascade. And don't worry about which complement protein does which. They were named in the order that they were discovered, not in the order that they actually turn each other on. So don't worry about the number assigned here. But the end result is always a membrane attack complex. And basically it busts through the cell wall and busts through the cell membrane, and it causes the cell to lyse. Now, in the process of turning on all of these different complement proteins, some of them actually will go off and create what we call an opsonization. So remember, we just said phagocytes have to be able to adhere to the foreign particle in order to ingest it. But some of them have things like a capsule, which keeps them from having affinity. So some complement proteins will actually attach themselves to the capsule and give phagocytes a little handle that they can grab onto. And so complement would be the opsonin, right? It's just a little coating molecule. Opsonization is the process of covering the bacteria with opsonins. And so that just increases the chances that phagocytosis can happen. The other thing is some of these complement proteins, once they're activated, will actually act much like a histamine molecule, which will trigger inflammation. And of course, the whole process of inflammation we'll talk about shortly. It's just going to increase the chances that phagocytes can come to the neighborhood where this is happening. And so it's all collectively going to help kill the bacteria that it comes in contact with. So again, inflammation is the response that our body has to any kind of tissue damage. So chances are, if you have tissue damage, especially if it's superficial tissue damage, those bacteria on the surface of the skin will get access into the body. And so we need to have a method for coming in and dealing with them as well. So while we think of tissue damage being repaired by inflammation, it's also going to make sure that there's an immune response right there at the location where there's a problem. So anytime you have damage to the tissues, you have things like mast cells in the tissue that release histamine, and histamine is going to be the trigger for this process. And so if you recall, histamine and other inflammatory chemicals will increase vasodilation in the neighborhood. And so this is going to increase blood flow to the site. This is going to invite white blood cells to the site where the inflammation response is happening. And so by creating this situation where histamine does two things, it vasodilates the capillaries nearest to the inflammation site, and this creates a gap in the wall as well. So we have both vasodilation and increased permeability, which does two things. It allows the fluid, what we would call plasma while it's in the bloodstream, gets called exudate when it seeps out of these interstitial spaces that are produced by inflammation. So this fluid that's oozing out into the neighborhood, if you get enough of it there, you'll have a visible swelling that we call edema, right? So edema is the result of the fluid called exudate that has entered the tissues. And so this does two things. It helps to isolate the problem, right? Picture if you're out here in the interstitial space and you're a bacteria and you are trying to get into that bloodstream, it's a lot less likely that you're going to be able to swim upstream against this fluid that's pushing against you. And it also, by having the gaps in the capillary wall, allows those white blood cells to actually crawl through the gaps and find its way into the tissue so it can go out and do phagocytosis. So if you recall this word diapedesis, we learned about that in A&P2 when we talked about blood cells. So this word diapedesis just means jumping over the capillary wall, and basically now they have access to the inflammation site, and they can go out and gobble up any foreign particles, or they can help get rid of cellular debris in the damaged area. But also this exudate will deliver everything that's necessary, right? We just talked about interferon and complement proteins, antibodies, nutrients, oxygen, everything that the tissue is going to need to repair itself. And so if all goes well during the process of inflammation, then tissue repair should be very efficient, right? You're going to have the collagen forming, and the epithelial cells will replace themselves, and hopefully that tissue will fully be repaired. So another process that the body can use to protect itself against foreign particles is fever. And so this is where the body temperature will be raised in response to chemicals that are generally referred to as pyrogens. So the types of things that can serve as a pyrogen would include things like bacterial toxins. So for example, C. diff and Staphylococcus infections, these types of bacteria secrete toxins into the body that are very powerful pyrogens. You'll always get a fever with those types of infections. If it's a gram-negative bacteria, if those cells are killed by any mechanism, that LPS that is found in the outer membrane, when it's released into the body, it actually will be interpreted as a pyrogen and will trigger fever. Interleukins are really important little messengers that are released by phagocytes, which of course are the best source of this kind of a message. If a phagocyte continues to encounter and destroy bacteria, as long as it keeps finding new bacteria to destroy, it keeps sending out interleukins. And so it's a way for those phagocytes that are out on the front lines to communicate with the brain that there's still an infection going on. Also, antibodies. If an antibody has bound to its antigen, we call these immune complexes. If these are still floating around, the hypothalamus will see them and recognize that, okay, the infection is still continuing on, and we want to continue to fight it with the process of fever. And so how fever is produced in the first place is when the hypothalamus sees those pyrogen molecules, it will trigger the liver to ramp up its metabolic activity. So recall that every chemical reaction releases a little bit of heat. And so by asking the liver to do a little more chemical metabolic activity, it's going to help raise the body temperature. And really, it's really the core of the body that gets warmer, because the other thing that's going to happen is it's going to cause the peripheral blood vessels to vasoconstrict. And by constricting peripheral vessels, it will force all the blood to the core of the body, which is how you get a higher core temperature. And so it might not make sense that if you feel like you're burning up, but at the same time you feel chilled, it's because your thermoreceptors are out in the skin. So if you've pulled the warm blood away from the skin, then your thermoreceptors are going to tell you you're cold, even if you're burning up at the core. And so because interleukin-1 is one of those really powerful pyrogens, as the bacterial load in the body decreases, right, as phagocytosis is effective, or maybe complement or something has come along to destroy those bacteria, as the bacterial load goes down, then the chemical message of interleukin-1 will go down, and it'll eventually tell the hypothalamus the war is over, we can reset the body temperature back to normal. And so this is one of those negative feedback mechanisms. So fever is a very beneficial process, but not in the way that most people think. It's not about raising the body temperature so high that it directly kills by sort of just sizzling up the microorganisms. What it does is basically make the body better at the immune response. And so one of the ways it does that, one of the most important things it does, remember, higher temperatures denature proteins. So the liver and the spleen have proteins that are designed to keep iron and zinc locked up, because free iron and free zinc is very toxic to organs like the kidney. And so by raising the body temperature, you cause these proteins in the liver and the spleen to sequester, or basically lock up iron and zinc and keep it out of circulation. And this is to keep those cofactors or coenzymes away from bacteria. So if you recall, for them to replicate, they need enzyme activity. Enzymes require heavy metals typically as the cofactor. So if these heavy metals, which are, by the way, the most common cofactors used by living things, if those are locked up in the liver and the spleen, wherever the bacterial infection is going on, they don't have what they need to replicate, and it basically buys time for the immune system to go after them. So by ramping up the body temperature, we also make other aspects of the immune system more effective. Remember that interferons are going to be produced by virally infected cells. Antibodies are little proteins that have to be made by our B cells, and those will happen more efficiently. Phagocytes and lymphocytes that are having to be made in the bone marrow in response to inflammation and other types of recruitment activities. Those cells are going to be not only made faster, but they're going to be able to do their jobs faster. And tissue repair will be more efficient. So, of course, fever is something that, unfortunately, at the first sign of fever, many people go running for aspirin or other fever-reducing drugs, which is really unfortunate, because if you let a fever ride, it's actually going to improve your immune response. Obviously, if it's too high for too long, that's a problem, because it will start denaturing proteins that can't be repaired. Fortunately, our cells are complicated enough or complex enough that we have what we call chaperonins, and these are little proteins that can go help refold important proteins that have been denatured. The brain, for example, has a lot of chaperonins because otherwise we would have febrile seizures every time the body increased its temperature just a little bit. Bacteria, fortunately, don't have chaperonins, and so there are some species that are very sensitive to temperature shifts because they can't repair their proteins. Historically, there's some evidence that doctors would intentionally infect people with malaria because that disease causes your body to go to really high fever in order to cure things like gonorrhea and syphilis before we had antibiotics. Now, of course, the person had malaria, but it's also a disease that an adult could survive. And if you get past those long-term infections like syphilis, then it was somewhat effective. There's some research now that shows if after a needle stick or some potential HIV exposure, if the patient is triggered or exposed to a very high body temperature, if you induce fever in that individual right after the exposure, then it reduces the risk of that virus ever finding and infecting the proper host cells. So there's a lot of research into whether that's going to be an effective post-exposure prophylactic type of treatment. So this is a nice little summary table of those first and second line defenses that we've been talking about. In the next chapter, we're going to talk a lot about the lymphatic system, because this is really where your third line defenses are actually going to do their job. And so if you recall what the lymphatic system is about, it's really about fluid recovery, right? So at the capillary beds, any fluid that leaks out of the capillary bed then gets picked up by the lymphatic system. And it's a one-way system, so it's a very slow-moving fluid. And as it moves back towards the heart, it will get filtered through lymph nodes. And so we think of the lymphatic system as a fluid recovery and a good place for immune surveillance, because those lymph nodes are going to be a place that we load up with white blood cells that know how to watch for foreign particles. And so we refer to lymphatic organs as primary if this is where the immune system cells are made or matured. So we're going to talk about places like the red bone marrow, where we're going to make our B and T cells, as well as all other white blood cells, and the thymus, where we're going to mature T cells. And then once they're mature, they will leave the primary lymphatic organs and go out and set up camp in what we call the secondary lymphatic organs. And this is where the actual fight is going to be, what we call the antigen challenge. It's going to happen in places like the lymph nodes, the spleen, and other tissues throughout the body, like the tonsils and Peyer's patches in the intestines and things like that. So we'll pick up on those on the next chapter. And so again, some of the chemical messengers involved in triggering immune responses are oftentimes called cytokines. Cytokines can be defined as somewhat like a hormone, except hormones leave a gland and flood the body and go talk to a variety of other cells. Cytokines are generally thought of as more of a direct message from cell to cell. And so think about things like interleukins that we talked about that triggers fever. Interleukins are really messages that go between one white blood cell and another. That's where the name interleukin comes from. We have inflammatory chemicals like histamine and serotonin. We even do have a hormone called erythropoietin that's considered a cytokine in the way that it talks specifically to the stem cells in the bone marrow to trigger them to make red blood cells. So you'll hear about more of these chemical messengers when we get to chapter 13. So that's it.